One of the major aspects of the continuity of living systems is the ability of cells to pass on a complete set of genetic instructions to their daughter cells after cell division. This inheritance of information is dependent on the ability of a double-stranded DNA molecule to copy itself, producing two identical daughter molecules. This process, called replication, occurs during the S phase of the cell cycle. In quick summary, the double helix is unzipped and each single strand acts as a template to construct a new strand. Because of the base pairing rules, this creates two new identical DNA molecules. This type of replication is called semi-conservative because half of each newly replicated molecule consists of old material and the other half is new. Replication is initiated at an origin of replication where enzymes called helicases unwind the double helix so that the strands are available to act as templates. Additionally, this unwinding occurs to make room for the required replication enzymes. Single-stranded binding proteins, or SSBs, associate with the unwound helix to keep it from reassociating with itself. Replication proceeds bidirectionally, creating two replication forks that move in opposite directions. DNA polymerase is the enzyme that actually adds new nucleotides and covalently binds them together after they have initially assembled themselves on a template strand by correct base pairing. The polymerase can only add nucleotides to a growing chain and can never initiate growth. Therefore, every chain must be started by the action of the enzyme primase, which lays down a short RNA primer. Since DNA polymerase can only facilitate growth in the 5' to 3' direction, and because of the anti-parallel nature of DNA, one strand must be replicated continuously, known as the leading strand, while the other grows discontinuously, the lagging strand. This fact can be verified by the presence of Okazaki fragments on the lagging strand, short pieces of DNA that must eventually be joined together by the enzyme ligase. DNA polymerase also has a 3' to 5' exonuclease activity that allows it to correct errors and to remove and replace RNA primers with DNA. When the process is complete, the two new identical daughter molecules have folded back into a double helical form, and one copy of a cell's genetic design information has become two.